Good morning students. Today we are going to talk about the dark reaction. This reaction discovered by F. Blackman scientist in 1905 and all biochemical reaction taking place during this phase worked out by Kelvin, scientist and its co-workers. Okay. Now, site of dark reaction. Dark reaction take place in the stroma region of chloroplast. So, stroma is the site of dark reaction. CO2 absorbed by plants from atmosphere is reduced or fixed in the form of carbohydrate, means sugars, and energy is used in CO2 fixation, which is obtained from ATP and NADPH2. From light reaction. It means dark reaction is fully light independent reaction. Second feature, it is fully enzymatic reaction. It is also known as biochemical reaction. It is also known as CO2 fixation reaction. So these are the features of dark reaction. Okay. Now, CO2 fixation may be take place by different methods in plants. So there are three methods for CO2 fixation in plants. First, Kelvin Benson cycle. It is also known as C3 cycle. Second, H and Slake cycle. It is also known as C4 cycle. And third, cressolation, acid metabolism. It means chem cycle. So there are 3 cycles in the plants for CO2 fixation. Firstly, we are going to talk about the Kelvin-Benson cycle. It means C3 cycle. It is discovered by Kelvin and Benson scientists in 1946 to 1953. Kelvin and Benson scientists used carbon-14 radioisotope of the using of CO2 in his experiment, and work carried on unicellular algae, for example chlorella, and second Senedesmus. Chlorella and Senedesmus are the example of algae. It means these are algae. And firstly, C3 cycles was discovered in the Chlorella and Senedesmus by Kelvin and Benson scientists using a carbon-14 radioactive isotope. Okay. In this algae, they observed that first stable compound in this phase is 3 carbon, which is PG, full form phosphoglyceric acid. PG is a 3 carbon compound. Due to this, name of this cycle is C3 cycle. In 1961, Kelvin and Benson were awarded by Nobel Prize for this experiment on chlorella algae and Synedesmus algae. Now we are going to talk about the C3 cycle in detail. This cycle is divided into 3 phases. First, carboxylation. Second, reduction. And third, regeneration. Reduction or reverse glycolysis, regeneration or formation of hexose sugar or regeneration of RUBP. RUBP is a 5-carbon sugar, full form ribulose 1,5-bisphosphate structure of ribulose 1,5-bisphosphate is first, second, third, fourth and fifth carbon. First and fifth carbon position phosphate group is added. The name is ribulose 1,5-bisphosphate. Okay, let's see. Cyclic process. Plants absorb atmosphere CO2 and this CO2 enter through stomata in the leaves of chloroplast. And CO2 is received by CO2 acceptor 6-RUBP. RUBP is a 5 carbon compound, full form tribulose 1,5 bisphosphate, and it acts as CO2 acceptor. Here RUBP react with 6 CO2 molecules and 6 water molecules. Then formation of 3PG, 3-phospho glyceric acid. Three-PGA is a 3 carbon compound, and this reaction occurs through RUBP, carboxylase enzyme. It means Rubisco. Rubisco help in the CO2 fixation. It means reaction of RUBP with CO2 and water molecules by Rubisco enzyme. Then RUBP convert into a 3 carbon compound which is known as 3-phosphoglyceric acid, and it is the first stable compound of C3 cycle. That's why Kelvin-Benson cycle is known as C3 cycle. Further, 3-PGA using of 12 ATP molecules of light reaction ADP plus inorganic phosphate. Due to breakdown of ATP molecules, formation of 12 ADP and 12 IP, inorganic phosphate released out from ATP. Then ADP is formed, and these ATP are obtained from light reaction, and this reaction occurs in the presence of tri-phosphokinase enzyme. Then 3-PGA convert into 1,3-di-PGA. It means थर्ड नंबर एंड फर्स्ट नंबर कार्बन पे फॉस्फेट ऐड हो जाता है. देन फुल फॉर्म 1,3-di-phosphoglyceric acid. Further, 1,3-di-PGA use of 12 NADPH2 of light reaction and convert into 12 NADP plus and this reaction occurs through triphosphate dehydrogenase. Dehydrogenase enzyme used in reductional reaction and kinase enzyme used in phosphate addition. Okay. Now 1,3-di-PGA convert into 1,3-di-PGAL. 1,3-di-phosphoglyceraldehyde. PGL and PGA are different. Full form of PGA is phosphoglyceric acid and full form of PGL is phosphoglyceraldehyde. Means PGA converted into PGL. It is the reductional reaction. Now phosphatase enzyme act on 1,3-di-PGA, then phosphate are removed from first and third number carbon of PGL and PGL convert into PGA. There are 12 molecules, 6 molecules of RUP, further 12 molecules of PGA, 12 molecules 3-di-PGA, then 12 molecules of 1,3-di-PGA. And these are 3 carbon compounds. PGL interconvertible into DHAP. DHAP is also 3 carbon compound. Phosphoglyceraldehyde also can be converted into dihydroxyacetone phosphate. Dihydroxyacetone phosphate. These are interconvertible each other, and this reaction occurs in the presence of isomerase enzyme. Further, 3 carbon of PGL and 3 carbon of DHEP and 2 molecule of PGL form fructose 1,6-diphosphate in the presence of aldolase enzyme. Aldolase is a condensed enzyme. It must the two compounds. Further, fructose 1,6-diphosphate convert into fructose 6-phosphate in the presence of phosphatase enzyme. Phosphatase enzyme but remove the phosphate. Phosphatase enzyme remove the phosphate from fructose sugar, first number carbon. Then fructose 1,6-diphosphate convert into fructose 6-phosphate. Further, isomerase enzyme act on fructose 6-phosphate and form the glucose 6-phosphate. Further, phosphatase enzyme also act on glucose 6-phosphate and remove the phosphate at 6th position of glucose. Then it convert into glucose. So it is the formation of hexose sugar in C3 cycle. And third step, PGL plus DHAP दोनों मिलके रीजनेशन भी कर सकते हैं. राइबोलोज 5-phosphate convert into 6-RUP. Then 6 ATP molecules are used and 6 ATP convert into 6 ADP plus IP in the presence of triose phosphokinase enzyme. Now 6 RUP to 3 PGA known as carboxylation, 3 PGA to 1,3-dipGA known as reduction or reverse glycolysis process, and from PGL and DHAP formation of hexose sugar and 10 molecules of PGL plus DHAP regenerate the ribulose 1,5-bisphosphate. So there are 3 phases completed and formation of hexose.